Brown Bag Lunch Program and is a collaboration between the HIV Prevention Trials Network, the HIV Vaccine Trials Network, and the Microbicide Trials Network. So my name is Rebecca Israel with the Black AIDS Institute, and I'd like to thank you all for joining. So just a few housekeeping before we get started. If you're having any technical difficulties, whether it's visual or audio, make sure that you have downloaded the Citrix software because some people may not be able to hear or see, and you have to make sure that you download the GoToWebinar software for that to work. We also want to encourage you to ask questions throughout the entire webinar. You can do that in two ways. The first way is to raise your hand. If you can see my mouse, it's right where my mouse is, the raise hand button. And then you can use the question box as well. So keep the questions coming throughout the entire webinar. And then this webinar is also going to be recorded. This is an hour-and-a-half long webinar, and so I know some people will be joining late or having to leave early. So we will be emailing the recording to all the attendees after it is finished. And so just to give you a brief background of the Brown Bag Lunch Program, this is our last webinar of the year, and I know that there are some people who are on the line who are not familiar with what the program is, and so I wanted to give you all a background of that. So the Brown Bag Lunch Program is a train-the-trainer webinar series that's focused on community education. So it's unique in that we don't just focus on the individual education of the person who attends the webinar, but instead we give the attendees the tools and technical assistance to host community events where they train their community on the topics that they learn during the webinars. And so I've listed for you some of the topics that we've gone over this year and some of the partners that we've had this year. And these pictures that are on the slide, I've included these just to give you a sample of one of the community events that our Brown Bag Lunch leader has done. And this is one of our stellar Brown Bag Lunch leaders, Linda Stringfellow out of Mississippi. And this event that she did was on creating safe spaces for HIV disclosure, and that was a collaboration with HPTN as well. And so we're grateful for all the hard work that our Brown Bag Lunch leaders have done this year, and we're encouraging everyone who's not a part of the program to apply for the program for the 2015. And so here's our agenda for today. First, we're going to have an introduction to clinical research, just to kind of set the stage for what we're going to learn about from the updates from the different clinical trials networks. And then we're going to hear from MTN, then Vaccine Trials Network, and then HPTN. And then we'll have a section for questions and answers. And again, we encourage everyone to send questions throughout the webinar, and then we'll try to touch on some of those after each presentation. And if there's extra questions at the end, we'll address those as well. And then we'll have a few announcements. We'd like to hear from you about your World AIDS Day events that you're either hosting or attending, and any other announcements that attendees may have. And so I'm going to get started, and I'll introduce to you all the first presenter. Her name is Georgette King, and she serves as HPTN's Senior Community Engagement Officer for the past 5 years. In that role, she's developed relationships with national and regional organizations with the goal of facilitating access to information about HPTN's research and encouraging the dissemination of that information within the communities served by those organizations. She administrates HPTN social media sites, so she's definitely the person to be in contact with for HPTN. And so if you're interested in learning more about how the community gets the clinical research information, Georgette is the person to contact. And so she'll be going over the clinical trials and intro to clinical research for us. And so, Georgette, I'm going to turn it over to you.
Hi, thank you, Rebecca. I'm going to give a brief introduction to clinical trials so we can go ahead and hear from our presenters from 3 of the networks. So if you would move to the next slide, please. So the networks that are represented today are part of the Division of AIDS, DAIDS, And during the webinar, you're going to hear from 3 of them. If you would click one time forward, please. These are the 3 that you'll be hearing from. Apologize for a little migration on the slide. It's HPTN, HIV Prevention Trials Network, the HIV Vaccine Trials Network, and the Microbicide Trials Network. The Division of Acquired Immune Deficiency Syndrome, DAIS, was formed in 1986 as an extramural division of the National Institute of Health, National Institute of Allergy and Infectious Disease, NIAID, and its purpose is to develop and implement the national research agenda to address the HIV/AIDS epidemic. Can you move forward, please? So I've been asked to give a little information about clinical research. And clinical research is generally defined as carefully conducted investigations that ultimately uncover better ways to treat, prevent, diagnose, and understand human diseases. The information I'm giving you is from the NIH website, and there are a number of resources there that you can use as you talk to communities about clinical research. Next, please. Now, we know there are different phases of clinical research, so I'm going to go through the phases. And again, the additional information you'll be able to find on the website that's shown at the bottom of the page. So a phase 1 trial is organized by researchers who are testing an experimental drug or treatment in a small group of people, usually a group, as it shows, between 20 and 80 people for the first time. And the purpose is to evaluate its safety and identify side effects. So a phase 1 trial is not looking to see whether a drug necessarily works for the purpose that is hoped for by the researchers. It's just to find out if it can be tolerated by people and if it's safe to use. When you move on to a phase 2 trial, that's an experimental drug or treatment that's administered to a larger group of people, usually between 100 and 300 people, to determine the effectiveness and to further evaluate its safety. So it's not until phase 2 that we're looking to see whether a drug actually serves the purpose that the researchers are hoping it'll serve. And again, there's additional look at the safety of the drug. Next slide, please. A phase 3 trial is now stepping up to a larger level. So it would enroll between 1,000 and 3,000 people, and it's a confirmation of effectiveness. There's also a lot of work done to monitor side effects, to compare that treatment that is being tested with a standard or equivalent treatment, and to collect information that will allow the experimental drug or treatment to be used safely. So that's a trial that has really stepped up once it's believed that the drug is effective. And the last phase that we're going to discuss, if you move to the next slide, please, is a phase 4 trial. After a drug is approved by the FDA and made available to the public, researchers then do more work to track its safety and to look more closely at a drug's treatment's risks, benefits, and optimal use. So those are generally the phases of clinical trials that you'll hear referenced. Next slide, please. Now, I wanted to show you a great resource that's available through the NIH website. And as I noted here, I'm only showing you excerpts from the slide set. So this is a slide that's available to download, and you can use this when you're having conversations with community members, or if within your agencies you are trying to get some basic information about clinical research. So Rebecca, if you just go through the next couple of slides. I'm not going to talk about these slides in detail. You can keep advancing, because again, this is a slide set that you can pull up on your own. Okay, this is the slide I'd like you to stop at. So this shows some of the resources that are available for you to download. This first page is a set of badges that you can add to your website. And again, if you click through these badges, you'll see additional information on all these issues. If you go to the next slide, please. And this slide just shows you some more resources that are available. You can print these as poster size, your smaller size. This is also where you would pull up the slide set that I'm showing you. If you go to the next slide, please. And there are volunteer stories that are available here. And each of these stories are really designed to discuss some of the concerns people may have about joining clinical research or to talk about experiences. So it's a good place when you're getting questions about why people would actually want to participate in clinical research. Next, please. NIH has also made quite a number of pamphlets available. So some of these may be good for you if you are working in a clinic setting and you want to have information there. So as you're discussing clinical research that's being conducted in your area with people, they can get additional information. And there's also information that's there really for the providers to give them a better sense of how to effectively refer someone to clinical trials, or why it may be a feature that they want to consider adding to their office setting, so that there's more information about how it may benefit patients or community members to have access to clinical research. And then there's just one more after that that I'll show you. And the final slide of the set really talks about how to locate research. We very often get questions about whether HIV research is being conducted by one of the DAIDS networks in a particular community, or if there's other HIV research that's being conducted. This is an excellent site to go to find out where research is being conducted. There's a lot of research that happens around HIV that is not part of the DAIDS network, so it may not be a study that we're as familiar with, but there may be work that's being done in your local area that you'd want to get involved with, or to find out about the results or the impact from the research that's being done. So this is a site that you can go to look up and see what's being done. And I think it's interesting to see the wide range of research that's being conducted and the different types of research that's being conducted. But again, this slide is a part of that master set from NIH that you can download to use within the community or doing a presentation to talk about what's happening with research. And this is a screenshot, obviously, from the NIH page, so you can go there at any time and look up this information yourself. And the final slide, please. And this is the closing slide from that presentation, and it's just a reminder that the purpose of the NIH is to turn discovery into health, and we will move on with the discussion from the three networks that are with us today to talk about the type of research that they're conducting and the future of research in those networks. So that concludes my presentation. I'll turn it back over to you, Rebecca.
Great. Thank you, Georgette. Before we go on to the Microbicide Trials Network, we did have a question from Julie for you, Georgette, and she said, can you tell us where in the phases are the confirmatory trials and the implementation trials?
Sure. That phase 3, I believe, is what you're referring to for confirmatory trials. And if one of my colleagues feels that that answer should be different, please jump in and let me know. And then implementation, that usually would happen in a slightly different format. So at the time that the phase 4 trials are going on, after they have reached that large scale up and a drug has been shown to be effective and safe, then another type of research would happen, which is what you're referring to as implementation research, where there's an opportunity to see just how the community feels about a particular type of intervention, or to see the best way of rolling it out. And in fact, HPTN study 073, which is looking at the use of PrEP with gay black men in 3 cities throughout the United States, would be an example of that type of research. And when we reach the HPTN presentation, perhaps Jonathan can say a little bit more about the way that implementation study is conducted.
I think Jim had a comment.
This is actually Wakefield. I just wanted to jump in, and I think Georgette wrapped it up pretty nicely, but a confirmatory test is really the phase 3 where we've had a hypothesis, phase 1, we've made sure we're moving forward in a safe manner, and in the phase 3 we are able to answer the scientific question. But as we move into phase 4 studies, that's when we're able to see how does this translate in practically in all of the communities where we work. But it's a great question. Thanks for asking, Julie.
Thanks, Wakefield, and that's for those who are on the call, another one of our colleagues from the HIV Vaccine Trials Network. So I don't see any other questions, but thank you for your responses to those questions. And so we're going to go ahead and move on to the Microbicide Trials Network. And our presenter for today is Clare Collins, and she's the Associate Director of Communications and External Relations for MTN, where she has focused her efforts on rectal microbicides research for the past 4 years. She has also worked as a fellow in health communications at the National Cancer Institute, and she holds a master's in women's studies and in health promotion and education from the University of Cincinnati. So I'm going to turn it over to Clare.
Hi, everyone. Can you hear me?
Yes.
Oh, great. Thank you. Good afternoon. Thanks to the Black AIDS Institute and HPTN for inviting me to speak at this webinar today. I really appreciate it. Next slide, please. So who the Microbicide Trials Network are, we're an organization that actually focuses on microbicides, which are products that could be gels, could be films, could be rings that are applied either during sex, around the time of sex, daily. So we're looking at these products in clinical studies and to see whether or not they might be an option for people for HIV prevention. If you look at the picture here, we're actually furthest along in testing gels, which are applied with the use of that sort of white applicator with the blue tip, and we're also farthest along in vaginal rings. So those are the studies that we're at. The vaginal ring studies are in the phase 3 stage that Georgette talked about, and our microbicides have been in phase 3 as well. And these other kinds of formulations are really just under development, but we're not in current clinical trials for some of those other modalities. Next slide, please. So our studies are conducted really all over the world. We span 4 continents, including the United States. We're in Malawi, South Africa, Uganda, Zambia, Zimbabwe, Thailand, Peru, and as I mentioned, the United States, and that includes a study in Puerto Rico as well. Next slide, please. In the United States, on this slide you can see a few of the cities where we're particularly located. Many of these studies are phase 2 studies currently. Some of them are phase 1. We hope in the future to have a phase 3 rectal microbicide study in the United States. That won't be happening for a while yet, but you can get a sense here where we're located. Next slide, please. So what we've done so far is we've had 14 completed trials. We have 9 that we're planning or in progress. You can forward all these on, Rebecca. We focus primarily on antiretroviral-based approaches in our microbicides. They're based on drugs and products that we know a lot about because they're products that, for the most part, have been used in HIV treatment. And as I mentioned, we're looking at these different formulations. We're looking at how we can deliver them. We're really interested right now in long-acting vaginal rings. We've also really significantly invested in rectal microbicides because we recognize the need for products that people could use during anal sex to protect against HIV. And we actually have also done some studies in pregnant and breastfeeding women because we're kind of looking at this as your whole lifetime. You know, what across an entire life, including reproductive life, what products could protect women and men. The other important part of what we do is behavioral science. So I think all the networks actually spend a lot of time on this. Next bullet. We really understand that human behavior is very important. You can have a product that you know medically works very well, but if people really don't want to use it and it's not presented in a way that's acceptable to them, then it really is not effective. Next slide. So I'm going to talk briefly just about some of our flagship studies. So we have a lot of studies in the field, but these are the ones that we've spent particular time on. The first one here is VOICE. Some may have heard of VOICE. It was a phase 2B trial, which means it's almost a phase 3, but not quite, although it was pretty close to a phase 3. It involved about 5,000 women in 15 African sites, and we conducted that between 2009 and 2012, and we actually reported results in 2013. So this trial was actually, well, actually I'll get into that a little later about what tenofovir and Truvada are, although many of you may know what Truvada is. Another one of our flagship studies is ASPIRE. This is a phase 3 study of the piperine vaginal ring, and we began this in August 2012. We're fully enrolled in this study, and it's actually being conducted in parallel with the International Partnership for Microbicide, the ring study. So both of these studies are sort of doing the same thing. And the reason why we wanted to do this was if we find out the product works and acceptable, we wanted to have 2 studies that show that so that then it could support the potential licensure of the drug. So we expect these results in early 2016. Next slide. And as I mentioned, we're very focused on rectal microbicides, and we have the first ever phase 2 study, MTN 017, that's testing tenofovir gel, which many of you may have heard of, but it's a reduced glycerin formulation of tenofovir gel. So it's a little bit more rectal friendly, which I'll talk about in a little bit. We're enrolling 186 men who have sex with men and transgender women in the U.S., Puerto Rico, Peru, South Africa, and Thailand. And we hope to have results in mid to late 2015. And actually, I want to say something really quickly that we just found out yesterday that all the sites have completed enrollment in MTN 017, so we're very excited about that. Next slide. So I want to go back to VOICE just for a minute so I can explain this a little bit more fully. So VOICE was looking at these 3 different products, tenofovir tablets, Truvada tablets, and tenofovir gel. And you can see in this photo here, tenofovir gel with the applicator. We wanted to know, which is effective, are they all safe, which will women use? And you all probably know that Truvada tablets are FDA approved now for use for HIV prevention. At the time we launched VOICE, that wasn't the case. So we wanted to compare all these things and see which would be more effective and which people would want to use. Next slide, please. So unfortunately, what we found in VOICE is that the daily approach that we were using in VOICE, so VOICE was using these products every day. We found that it didn't matter if it was a gel or a tablet, the women of VOICE did not use the product. Most of them didn't. So as a result, no product was effective. We also found that younger women and unmarried women were least likely to use the product, but were most likely to acquire HIV, which is obviously a concern. HIV incidence was 5.7%, so this was twice what we estimated it would be. So that was not something that we wanted to see, but that was the reality of the situation in many of these countries. And ironically, study retention was very high, 91%. So women stayed in the study, but they weren't using the product. And we didn't find any safety concerns with the product, which was a positive. On the next slide, please. So what we wanted to really get at here is after VOICE, we wanted to launch this behavioral sub-study. Although we were doing behavioral measures throughout voice, we really wanted to get at this issue about adherence and why did so many women not use the products? What was it about the products? What was it about their lives? You know, what could we learn from this? So we involved voice participants at 5 sites in Uganda, Zimbabwe, and South Africa. So we had a total of 15 sites. And again, aimed at better understanding this discrepancy between their reported use and their actual use. So we did this in 2 parts. We had part 1 where we had 88 women. After they exited voice, we did in-depth interviews. And then we did part 2. So we took 127 women who were on the active product, which that means they were on the actual product, not the placebo. So there was also placebo product involved in this study. There was a placebo gel as well as a placebo tablet. So these were all women who were on the real products. What we did is we presented them with results of their own blood tests to look at the drug levels. And we kind of used this as an icebreaker so we could have some in-depth interviews and focus groups to really understand the reasons why they did not use the product. We were kind of getting at this, you know, what are the contextual issues going on in women's lives that may have affected this? Are there any trial-specific issues that made it difficult for them to use the product? Next slide, please. We have really creative behavioral researchers on our team. So the way they wanted to describe this to women, because I'll back up for one quick minute. It was interesting that a lot of women, when these results were presented to them, especially the women in the lower adherence group, they were really surprised and in disbelief that the blood test showed that they didn't have any, little if any, product in their blood test. So we had to really get at this in a way where we weren't blaming women, we weren't saying, you know, you're at fault for not doing this, but we really wanted to have a real honest and frank discussion about some of the issues that they found involved in this. So we used this teapot tool to sort of show a visual of, you know, here's what a person that's a high adherer would look like. Here's an inconsistent, here's a low. So if you look at this slide, you can see the cups represent the drug at a visit. So looking at high, you know, all those cups are filled, and then in the second row there, only one isn't filled. So basically, they missed one day, but they were using the drug very consistently. And then the row depicts the pattern of use. And then the teapot is just the product use level. So you see the high adherers, you see the inconsistent. So, you know, there was drug in the blood one day, then none the next, I shouldn't say day, I should say this was done over the course of time, but you get the sense that it's inconsistent, that some days there was some points they came to the clinic, there was drugs, sometimes there weren't. And then with the low use, there was really hardly any drug in the blood at all. So that was the way we started this icebreaker conversation. And then we also had this theme card tool. So once that conversation was had, we wanted to really ask the women, you know, to what were some of the issues that they found really challenging in taking these drugs daily. So our researchers sorted the 20 cards into sort of themes. They rank ordered them. Then they had the women discuss the themes and identify any themes that weren't listed. So it might be hard to see the slide here, but some of the things were, you know, I was too busy to take the products every day. I had to hide when taking my products. I don't need to take them. I'm not at risk for HIV. Someone told me not to take the product. So it's sort of like, this was individual. So you're talking to women about, you know, tell us what was relevant to your life and which of these cards resonate for you. Next slide, please. So other than, you know, the surprise and disbelief, you know, some women came to accept the results once they were showed, some didn't. But what we found is providing the drug level results really led to some candid discussions and really getting at those adherence challenges. We found that the theme cards were pretty similar across the drug level groups. A lot of them were influenced by what they thought of the products, that there were side effects and harm. There might have been a pull to join or stay in the study that was perceived by the women, and provision of support, which could have been community support, which could have been partner support. As you can see here with the low drug, there were a couple differences in the low drug level group and the high level adherence group. So in the low drug, those people that really weren't taking the product, they picked out those themes related to fear of the products and side effects. They felt like other people's attitudes about what they were doing played a role in that, and negatively influenced by product use. For the high drug group, they generally said they were better able, we generally found they were better able to overcome the challenges. So they had some personal strategies that really worked, where they were able to continue to do what they wanted to do. I'll quickly just kind of read you a case study of one of the women from Uganda. She was 34, married and living with her husband who supports her financially, and she was in the low drug group. She said basically why she joined and stayed. She said, well, you know, I wanted to prevent HIV. I wanted the reimbursement from being involved in the study. I kind of felt bound by consent, or that the study staff could track me. Her top theme cards related to side effects from the product, her partner not liking it, and believing that the products couldn't prevent HIV. Why she said that she didn't use the products or used them very infrequently was she felt like there was an itching or a rash. She disliked the feel of the gel. She said it was cold, it leaked, there was too much of it. She feared the gel could harm her uterus, possibly cause cancer. And the way she said she used the gel is she said, I applied a little gel outside of my vagina for protection. But she was considering this whole fear issue, her partner's dislike of it during sex, so she tended not to use it during sex. And she said even after sex or urinating, she really felt very uncomfortable if she had some in and didn't wash it out right away. She said the way that she disposed of the gel is she gave some to her sex worker friend, and she expelled extra gel into a water bucket and poured it into the toilet. So that was just one sort of idea and case study. So in summary, we basically found this daily use of these products wasn't the right approach. Young unmarried women used them the least. And there was a lot of fears about the products and side effects, which were really fueled by peers, relatives, and community. And those are the most common themes for product non-use. So what we're getting at at MTN now is really the need for products women will want to use. So there's all these reasons why they didn't want to use it, but at the end of the day, they didn't use it. So we're really thinking of, we're already involved in a trial and exploring long-acting methods, and as I mentioned before, Aspire. And we're also interested in products that could also provide contraception. Next slide, please. And you can forward on to the next slide. So this is our study, Aspire, a study to prevent infection with a ring for extended use. So why a vaginal ring? Well, we find that they're pretty flexible. They fit high up in the vagina where they can release medication slowly over time. So instead of having, like the women did in voice, they have to use an applicator and apply a gel or take a daily tablet and remember to do that, this can actually stay in place during sex for about a month at a time. And a lot of women, not all in all countries, but women generally are more familiar with a vaginal ring product because we do have them in the U.S. and elsewhere for contraception and hormone replacement. Next slide, please. So again, Aspire is a product that is more long-acting, so it's replaced once a month. It might be easier for some women to use, and we're actually looking at a drug called dapivirine, which is different than tenofovir or tenofovir with emtricitabine, which is Truvada, the one that's approved by FDA. And we really want to do this so that if we find it's effective and acceptable, that we could actually license this ring. But of course, there's going to be other data that's important too. Next slide. So we're doing some supporting studies of the ring. One is a study in adolescent girls. We're conducting that in 4 cities in the United States. And another study is we're looking at the ring safety in postmenopausal women, and that's in 3 cities in the U.S. And we're pretty far along in recruitment for the postmenopausal study. It's been much more challenging to recruit for the adolescent study, but we're still on our way to do that. Next slide. I want to change gears for just a minute and talk about MTN 017. Next slide. So this is our rectal microbicide study. And just real quickly, since before I started doing this work, I really didn't know that much about the difference between the rectum and the vagina and the tissues. So it sounds funny, but it's really actually an interesting biological issue that makes people more at risk for HIV through anal sex. So one of the issues is, as you can see in the picture here, the vaginal mucosa is much thicker than the rectal mucosa. And what that is that's the barrier, the outer barrier of the rectum and the vagina. And so since the vagina is the rectum is one cell thick, it really makes a more fragile barrier. So, you know, anal sex is a really common behavior. It's in both men and women. I think people always associate it with men who have sex with men, but women practice anal sex in their relationships as well. And, you know, people talk about condoms. You know, why do you need rectal microbicides? We have condoms. Yes, we do have condoms, and they work very well if they're used consistently, if they're used correctly. But as we know, a lot of people either can't use them or don't want to use them, and you have to have a partner who agrees for that use. So you don't really control that use yourself. Vaginal microbicide research is farther along, so our first step was, well, let's just test the rectal safety of a vaginal microbicide. Next slide. Again, I've pretty much answered this. You know, why rectal microbicides? Well, Truvada is wonderful, and we're all like thrilled that we have this option to prevent HIV in the form of a tablet. But it might not be for everyone. And the idea of a rectal microbicide is you could have drug absorption in the body lower because you're not taking a pill that goes all through your body systemically, that you're using something in one place. We also feel like since lubes are often used during anal sex, maybe as a lube, microbicidal gel might have more of a chance of being accepted differently than the vaginal microbicide was in voice. Next slide. I'll go through these really quickly. These are just our previous studies to where we got to MTN 017. So as I mentioned, we first tested the vaginal safety of the rectal safety of the vaginal tenofovir gel. We actually found it caused some side effects, and even though it inhibited HIV in tissue samples, which is great, people didn't like the side effects. And if you have side effects in a product like this that's supposed to prevent HIV, people just aren't going to want to use it. I would not want to use something for prevention that caused side effects. So then we decided, since these side effects were related possibly to the glycerin that's in tenofovir gel, let's reduce the glycerin and then let's see what happens. So we did the follow-up study, MTN 007, and we found it was actually safe, much, much better tolerated than the vaginal gel version. Next slide. So now what we're doing in 017 is we're actually looking more at the reduced glycerin formulation of vaginal tenofovir gel. We're really looking still at the safety, so this is only a phase 2 if you go back to Georgette's introduction about the different phases of trial. So we're not at effectiveness yet. We're looking at safety, but we really want to find out about how does it compare to Truvada, how does it compare if you use it daily to if you use it before and after sex. You know, basically, what do people prefer? Do they like using it? Which regimen do they? Some people might want to use something daily because then they remember it every day. Other people say, hey, I'm not thinking about this until right before I'm planning on having sex. So I want something that's associated with sex. And would they use it in the future if it was available? So next slide, please. So we're looking at the safety data, and provided it's safe, our goal is to move on to an effectiveness trial. And I would like to, as I mentioned, announce that, you know, we're fully enrolled in MTN 017, so we're excited. We're on track for the completion of the study in mid-2015, and hopefully results between, well, hopefully by late 2015. So our goals for a network moving forward are we really want to demonstrate this effectiveness of the vaginal ring that I talked about, and we're already planning for a follow-up trial to the ASPIRE trial of the vaginal ring called HOPE, the HIV Open Label Prevention Extension. We've done a series of community consultations on that trial, and we're currently in the planning of that. And our idea is, you know, we finished enrolling MTN 017, completing the study, and then planning for a phase 3 trial of the reduced glycerin gel as a rectal microbicide moving forward. We're also interested in what we're calling second-generation rectal microbicides, so other products besides tenofovir that could be used as rectal microbicides. And we're interested in in the future of multi-purpose technologies. If anyone's heard of those, it's an awful word for them, but basically it's like combination products. So can we do some kind of combination rings that provide HIV protection as well as contraception? So those are the things that a lot of networks actually are thinking about and talking about in a lot of research groups. So we're excited about the possibilities of those. Next slide, please. And just keeping in touch slide, please feel free to follow us on Twitter. This is our website. On our website, especially under the news feature, you can access our news releases and statements. We have a lot of backgrounders and fact sheets on our work, and we also have all of our studies listed. So if you're interested in looking up any of those studies, they're all there. And these are the contacts: myself, Jonathan Lucas, who you'll be hearing from later, and Lisa Rossi. And lastly, just our acknowledgement slide about our funders. As Georgette mentioned, we're funded by NIAID, and we're very thankful to have the funding and to be able to speak to all of you today. Thank you very much.
Thanks, Claire. We do have some questions, but in the interest of time, we're going to hold those until the end. So Julie and I think Letitia asked questions, and so we'll definitely get to those questions after we hear from the other two networks. So I'm going to introduce you all to Jim Maynard, who is going to be speaking about the HIV Vaccine Trials Network, and he is the Director of Communications and Community Engagement for HVTN. Which is headquartered in Seattle. He holds a BS in biology and a Master's of Divinity from Eastern University in Philadelphia, and he has worked in the field of HIV prevention and community engagement for over 20 years. Before moving to Seattle, he worked at the Fenway Institute in Boston, directing community engagement and recruitment at clinical trial sites for HPTN, HVTN, and MTN. And he also ran 2 nationwide LGBT helplines for youth and adults. So I'm going to turn it over to Jim.
I see I'm unmuted now. Thank you very much for tuning in. My apologies for writing such a long introduction for myself that you had to read through. I'll make sure not to do that in the future. But very quickly, I'd like to introduce a special friend and coworker who I asked to sit in with me. His name is Steve Wakefield, and he's the Director of External Relations at the HVTN. He's actually been here for the entire history of the VTN, which was started 13 years ago. So I thought it would be great to have him as a resource in the room, as it were, to jump in on slides if he chooses to, but especially to be here for Q&A. So my job is to talk about vaccines. My goal is always a simple one, to talk about vaccines without putting you to sleep. Let's see if we can accomplish that. Instead of saying next slide, why don't I say just click to make it easier? How's that? Click.
Sounds good.
And there we go. So I just want to start off talking about vaccines for a minute. Now, when you hear the word vaccine, what do you think? I mean, honest and truly, click. Do you think of childhood vaccinations, which most of us had, and we had kids? Do you think of an annual flu shot that I'm hoping many of you got? I got my annual flu shot. Or do you think of big, sharp needles? Click. That's what most of us think of, isn't it? We think of big, sharp needles. Click. But it's funny. When I think of vaccines, this is actually what I think of. Believe it or not, that's a picture of my dear old dad when he was a little boy living on the farm. He was one of 11 kids. They were basically Irish potato farmers. And my dad is gone now. He died at 85, so this is a very old picture. But it was back in the days where a horrible disease was out there. The parents lived in fear of their children getting, called polio. My dad contracted polio, and he became crippled for 1 year. He couldn't walk. I love the story that my grandpa built him a little cart and attached a goat to it so that he could race around the farm and help his brothers collect the potatoes. This was a horrible disease that killed so many young children and left so many more crippled for life. My dad was one of the lucky ones. Click. Enter his grandkids, Sam and Jake, my sons. My sons, by the way, are both bigger than me now, many years later. But you know, we didn't have to worry as parents about polio, about what their grandpa got years before, because of something wonderful called research and vaccine development. Scientists, pharma, community members worked very, very hard to eradicate and defeat this disease. And the way it was done is the most effective way in public health to stop new infections. By and large, is by finding a vaccine to stop it up front. Click. Click. There we go. So what's the HVTN? I'm representing the HPTN, as is Wakefield. We're an international collaboration of scientists, trial sites, community reps, working with industry and government in the global search for an HIV vaccine with the goal of speeding up the development and of testing new HIV vaccine candidates. Click. HPTN is supported by a cooperative agreement with the National Institute of Allergy and Infectious Diseases, which is part of the NIH, part of the U.S. government. And our goal, our job, our task is a very simple and a very focused one: an effective vaccine against HIV. Click. Click. So what is a vaccine? I'll blow right through this because I'm guessing you all know what a vaccine is. A vaccine is a substance that teaches the body's immune system to recognize and protect against a disease agent. It's basically fooling your body. It's putting something into your body that looks like something that causes disease, and in that way your body can prepare well ahead of time to the day where if it does have to come up against that disease, it already has the little soldiers built that can beat it down very quickly. Next. So here's the impact of vaccines in the U.S. You can see what a huge difference it's made. Look at any one of these. I mean, look at something like smallpox. It used to infect over 48,000 people per year in the U.S. And now it infects how many? 0. 100% eradication of the disease here. The same with polio. You know, there's always a handful of polio infections in the world, 3 or 4 or 5 every year. So they're still looking to fight down those very fast cases. And then eventually that will be totally eliminated as a disease in the world as well. Vaccines have made a huge difference in public health. But how do we get to the vaccine? Next. It's years of hard work. You'll see here the number of years it took to develop each of these vaccines that have made a big difference. Polio, the vaccine that I wish my dad had had, took actually 47 years to develop. I just returned from visiting some of our sites in Africa. I was very thankful to have my yellow fever vaccination at the time, as I went into a country, Tanzania, that has active yellow fever. But you'll see how long it took to develop that as well, 35 years. Now let's move down to HIV. We have been working on the HIV vaccine for 30 years, and we still don't have one. Next. Margaret Heckler was the Secretary of Health and Human Services in 1984. That was the year that they figured out finally what the cause of AIDS was. They discovered a small virus, which they named HIV. And after that discovery, she made a very public announcement saying within 2 years we would have a vaccine to stop this horrible disease from spreading. Next. And we're working on it 30 years later. Can you imagine if we had developed a vaccine in 1986, how different the world might look right now? How much healthier the world would be right now? So I think it's a valid question. It's a question that any scientist can ask, community members, advocates, people that have lost those they love to HIV. Why is it taking us so long? Haven't we learned how to make vaccines better? Shouldn't the process be picking up and increasing over time to get vaccines faster and faster? Well, we have some unique challenges with the HIV virus, and let me talk about just 3 of them. One is that it has an extremely high mutation rate. It mutates so quickly that even after we have shown the body what it might look like, it changes so it doesn't recognize it. It also has the ability to hide its protein surface. The human body only recognizes foreign proteins. It doesn't recognize other foreign substances such as sugars or fats. HIV has the ability to fold in on itself and hide and have an outer surface of sticky sugars, so the body doesn't recognize it. And last but not least, the HIV virus destroys the very cells meant to protect against it, the T cells. What it comes down to is next, we're in a marathon. We're not in a sprint. Margaret Heckler imagined that it was a sprint. In 2 years we would get there. We discovered that no, it's a marathon. It's a long haul. And that's why I just want to take a minute here to praise the other networks doing the groundbreaking research. MTN and PTN are working on products to get them in the hands of people now. I'll admit that they're a lot closer to getting licensed products out there than getting a licensed vaccine that will take a few more years. Even after we get a vaccine may not be for everyone. And that's why we need a toolbox of prevention modalities. And that's what our sisters are working on as well. So, okay, where are we right now in development? Over 30,000 volunteers have taken part in trials so far. The HVTN currently has 20 open vaccine trials. And you know, to date, we've done 47 HIV vaccine trials. 54 vaccine trials. But only 5 of those trials have been efficacy trials. You'll remember from that great introduction from Georgette that a lot of trials are called phase 1 or phase 1-2 trials, where we're just testing it on a handful of people to make sure there's no bad reactions to it, that people can tolerate it, and that it's producing the correct cells in your body that would be protective. Only a very few vaccines ever get to the point of actually testing them to see if they work. And there's good reasons for that. One reason is that instead of testing it on hundreds of participants, you need to test it literally on thousands of participants. These are real human beings spending hours and days and months of their life. And we need to be careful when using that wonderful human resource as well as the millions of dollars. So we've done 5 trials so far. 4 of them did not work. Or I should say they didn't result in having an HIV vaccine. Only one of those worked. It was a trial ran by the U.S. military in Thailand called RV-144, testing a vaccine on 16,000 participants. They released the results in 2009, and the great good news is that we finally proved that we could develop a vaccine that would be protective. There had been a period in those years where some scientists and some advocates have started to worry, you know, it's not possible. But in 2009, the good news was, yes, it is. But after 2 years, we saw that it only protected 3 out of 10 participants. So it's too low to actually license because it wouldn't protect enough people. But we also discovered, click, that after 1 year, it protected 6 out of 10. What that meant was that it had a good protective factor, probably high enough to license at 60 to 70%, but that it didn't last. If we could only make it more powerful, more effective, and if we could make it last longer, then we'd have the vaccine we've been working 30 long years for. Next. So where do we go from here? Okay, this is the exciting part. It's all exciting, but this is today's exciting part. A group of people gathered together to form what was called the P5, the Pox Protein Public-Private Partnership. Just after 2009, the results came out. This is a consortium of folks like us that actually do the trials, pharmas that work on the drugs, funders, governmental agencies getting together saying, okay, this is what we're going to do. We're going to take the exact same vaccine combination that worked in the Thai trial, just didn't work long enough, and we're going to see if we can boot it up. We want to see if we can make it more effective. We want to see if we can make it last longer. So the goal is to get a safe, effective, and licensed vaccine to stop the spread of HIV. Next. Very quickly, how do we do that? The how-to is always the trick, isn't it? Well, scientists, and by the way, I'm not a scientist, they have different ways they're going to try to boost up the response from that to make it last longer and stronger. One way to do that is to switch out something called the adjuvant. An adjuvant is a chemical that's added to a vaccine, and simply enough, it boots it up. It makes it work better than it would all on its own. Almost all vaccines that you've had, including your once-a-year flu vaccine, has an adjuvant in it to make it work better in your body. Can you click that for me, please? I have a terrible memory. I got to look at it. So changing the adjuvant, we're changing it out to a different one. It's called MF59 that we found in other trials actually works a lot better than the alum that was used in 44. We're also going to put new protein inserts into that vaccine that will mimic what you find in HIV. More of these inserts hopefully will make it work better and it'll work broader. And last but not least, we want to add a boost, an additional shot, not just at first day, 3 month, and 6 months, but then adding one at 12th month. Next. Next. Oh, now the button's stuck. Okay, so this is what we're doing. This is not pie in the sky. This is not we're going to do a trial someday. This trial starts in January. We have something called the Product Development Track, which will happen in South Africa. We're going to start in January with HVTN 100. LVAC protein is the combination, the two vaccines that were used in RV144, but again, we've booted up and we've altered. We're going to do a phase 1 trial, 1-2 trial. Again, it's going to be a very small group of people, 252 HIV-negative, low-risk men and women, spread out over 4 sites in South Africa. We plan on opening it in January 2015. We want to test safety, and we want to see if we're producing the proper cells in the people's immune system. If we get a good, strong response in that, then we get into HVTN 702. Again, it's going to be the exact same products, but it's going to be a phase 3. This will be the 6th efficacy trial the world has ever seen. We'll enroll 5,400 people at higher risk for HIV in South Africa. Hopefully that will begin in the 4th quarter, the last quarter of 2016. So we're hoping this is what we call the licensed track. But by that time, actually it takes a while to enroll people. But sometime down the line from there, if we get the results that we're looking for, we can move forward to a licensed vaccine to stop the spread of HIV in the country and the world which is most impacted by HIV right now, South Africa. Next. And immunogenicity and efficacy. So we gave a name to this. There's actually going to be some sister trials happening at the same time called the research track. There'll be a half dozen other trials. They'll be testing slightly different products, different adjuvants, different delivery systems and ways of giving the vaccine, because we want to plan ahead for what we think of as the second generation vaccines. We're already planning on having one vaccine that works, but also doing the research that we can improve it over time. We decided to call all these vaccine trials together in Southern Africa. It'll include 5 additional countries in Southern Africa. Uwambo. Uwambo in Shona means opportunity. In Kosa, it means a journey. We're looking at this as a journey of opportunity, working with the people in Southern Africa, moving forward with vaccine development, and looking towards that day, hopefully in the not-too-distant future, when we're going to have a licensed vaccine like polio, like smallpox. Someday I want my grandkids to ask me, "Gee, Gramps, what's this thing about AIDS that I just heard about in my history class?" Next. Click. Thank you to everyone who helped me in this presentation today, especially to Steve Wakefield. It's appropriate I have him sitting here with me as I stole most of the slides from him. Click. And now I'm done. I assume you want to wait and hold off on questions till later, correct?
Yes. So we're going to hold off on all the questions. And so I'm just going to go ahead and move forward into HPTN, but keep the questions rolling in via the question chat, and we'll definitely get to them after Jonathan finishes his talk. So our last presenter for today is from HPTN, HIV Prevention Trials Network, and Jonathan Paul Lucas is a public health educator whose experience in community health in the community health arena spans almost 2 decades. He is the community program manager at FHI 360, and he works in the capacity of HPTN, MTN, and IMPACT, which are all 3 different clinical trials networks. Jonathan focuses on clinical research management, community engagement, and capacity building and advocacy of HIV, AIDS, public health programs, and research. And this is both domestically and internationally. He holds a master's and bachelor's in public health from UNC Greensboro. I'm going to go ahead and turn it over to Jonathan.
Hello, everyone. I'd like to first thank the organizers of this webinar for the opportunity to speak on behalf of the HPTN 077 protocol team on the implications of injectable cabotegravir. I'm sorry, I had a brain freeze right there. As a future option for HIV prevention. So next slide, please. This presentation will provide an overview of both the HPTN and HPTN 077, and it will also demonstrate how an injectable PrEP agent might improve product adherence. Next slide. So I'll begin with a very brief overview of the HIV Prevention Trials Network. Next slide. So the HIV Prevention Trials Network is a worldwide collaborative clinical trials network that brings together investigators, ethicists, community, and other partners to develop and test the safety and also efficacy of interventions that are designed to prevent the acquisition and transmission of HIV. HPTN studies evaluate new HIV prevention interventions and strategies in populations and geographical regions that bear a disproportionate burden of the infection. Next slide, please. So the HIV Prevention Trials Network research agenda is focused primarily on the use of integrated strategies, the use of antiretroviral drugs, which includes ART, or antiretroviral therapy, pre-exposure prophylaxis, as well as interventions for substance abuse, behavioral risk reduction interventions, and then also structural interventions. Next slide. So I'd like to talk just a little bit about the implications of an injectable PrEP. Next slide. So there have been several studies that have either recently been completed or are underway in quite a few countries involving more than 20,000 people with diverse HIV risk behaviors. All of these studies use 1 or 2 drugs as PrEP, either Viriad or Truvada. One of the most notable was the IPREX study, and this is a clinical trial, a large multi-country research study examining PrEP among men who have sex with men. And this found that daily oral use of Truvada provided an average of 44% additional protection for men who have sex with men who received a comprehensive prevention package, HIV prevention packages of services. And the level of protection varied widely. It depended on how consistently participants used PrEP, with significantly greater levels of protection among those who adhered well to the daily regimen. So among men who have sex with men with detectable levels of the medication in their blood, the risk of acquisition was reduced by more than 90%. The open-label follow-up to the IPREX study, the IPREX-OLE, found that the overall PrEP effectiveness of 50%, but 100% in those persons who took 4 or more doses a week that was measurable in their blood. Next slide. So there are 2 other research studies that are among women, FemPrep and Voice, and Claire spoke at length about voice earlier, and these found that participants provided PrEP did not experience any protection against HIV. An analysis of blood level data showed that very few of the women assigned to use the active drug actually took those pills as directed, and this indicates a lack of adherence, and this was the major contributing factor to that lack of effectiveness that was shown in both of these studies. The level of protection for PrEP varies widely depending upon how consistently participants use PrEP, with significantly greater levels of protection among those who adhere well to that daily dosing regimen. And other than low adherence, there don't seem to be any factors that have been identified that appear to influence the effectiveness of PrEP in reducing the sexual transmission of HIV. And this supports the notion that PrEP may only reach its full potential with agents that do not depend on daily pill taking. Next slide. So an exciting scientific advance is the formulation of new drugs in long-acting, slow-release injectables. And this has significant implications for patients who have achieved therapeutic success on an oral medication, but they need to maintain this over many years. So for HIV uninfected persons who are at risk for acquiring HIV, this might be a better option than taking pills daily. So the evaluation of cabotegravir, I'm never going to get this word right, and I really practiced it, as a long-acting injectable is at its early stages of clinical trial development, and we're trying to determine if this drug is safe and well tolerated before we test it to see if it's effective. So this particular study is a phase 2 randomized placebo-controlled study, and again, it's evaluating safety, tolerability, and it's looking at how the drug acts when it's in the body. And so study volunteers will be randomly placed into one or two groups. One group will be giving pills and injections that contain the drug, and another group won't get the drug. So there'll be 132 volunteers who get the drug, and then 44 volunteers who don't get the drug. Next slide. So HPTN 077 will enroll men and women in 8 cities in Rio de Janeiro, Brazil, Lilongwe, Malawi, Durban and Johannesburg, South Africa, Chapel Hill, North Carolina, Los Angeles and San Francisco, California, and then also Washington, D.C. Next slide. The study will answer several questions, including but not limited to these: are oral and injectable cabotegravir safe and tolerable? And how is the drug absorbed, distributed, and metabolized? Will research participants' sexual practices change as a result of getting the injections? And how much of the drug is in the blood of volunteers to become infected with HIV? Or for those who seroconvert, how often does HIV drug resistance occur? Next slide. So recruitment for the study is anticipated to take place over a 6-month period. And to enroll in the study, volunteers must report behaviors that indicate that they are at low risk for HIV infection. This is because we don't have very much safety data related to this particular drug. We don't know if it works. We don't want to enroll people who are at increased risk and they think that there's some benefit from this drug, because there may be no benefit. We don't know. All we have right now is animal data and phase 1 data. Volunteers must also be at least 18 years of age and able to provide informed consent. And volunteers who enroll in the study should not participate in any other biomedical prevention study, share or sell their tablets, or at any time stop using condoms. Next slide. So participants in the study will visit the clinic up to 19 times during the study, and at each visit, participants will receive a physical examination. They'll discuss concerns or drug side effects, and also receive HIV and STD counseling and testing, and they'll be offered condoms. So next, click again, and one more time. Perfect. So this is going to provide just an overview of what that study visit schedule is going to look like. So there in the blue, the participants are going to receive oral tablets for 4 weeks, and then they'll be followed by a 1-week washout period. Now this is to make sure that anyone who's on the tablets doesn't have severe adverse events. Because once you get that injectable in you, that may be with you for up to 3 months. So we want to make sure that there are no, outrageous adverse events. You don't want to have migraines for 3 months, or you don't want to have a severe rash on your arm that would last for 3 months. So if everything works out okay, participants are going to move into that yellow section. They'll receive an intramuscular injection at 3 time periods over a 6-month period, approximately. Can you click again, please? So participants in the active arm will be followed for 52 weeks after their last injection. And participants in the placebo arm will be followed until their last injection, or until the last participant in the active study completes their week 41 visit, whichever comes first. At which time the study is going to be unblinded, and then they're going to assess the primary endpoint, those study questions that I talked about earlier. And when all the procedures related to the unblinding are completed, participants in the placebo arm aren't going to be followed anymore. This study is going to last approximately 2 years, and this will include any of the closeout activities. The next slide. So I'll close with some final thoughts. Next slide. I think the main thing is that the HPTN has a strong commitment for quality research, and that PrEP is just one component of their very diverse research portfolio. And the science that they're doing with a number of their PrEP studies, both injectable and oral PrEP, it's going to inform the implementation of future PrEP programs and also future PrEP research studies. Next slide. This next slide just provides my contact information. If you'd like to get additional information on the HPTN, you can do so on our website, www.hptn.org, and you can follow us on Facebook and Twitter at HIVPTN. Next slide, please. So I'd like to close by thanking our funder. The HPTN receives its funding from 3 NIH institutes: the National Institute of Allergy and Infectious Diseases, the National Institutes of Mental Health, and then also the National Institute on Drug Abuse. I'd also like to thank the previous presenters as they provided a wealth of information as it relates to the HIV Vaccine Trials Network, and then also my partner in crime at the MTN, Clare Collins, did a very nice job providing an overview of the MTN. And now I'd welcome the opportunity to answer any questions that anyone has. Thank you.
Thank you, Jonathan. And thank you to our other speakers, Clare Collins, Jim Maynard, and Georgette King, for all presenting on their various networks. And so now we're going to open questions for everyone. And I'm going to start with the questions that people have already submitted. But if you have new questions coming in, please either use the raise the hand button and I'll unmute you so that you can ask, or continue using the questions chat box, and then I'll facilitate that as well. So we have a few questions that are already kind of in queue, so I'll go ahead and address those first. So we have one question about who participates in clinical trials, and we have a question that wants to know whether people in clinical trials are people living with HIV and AIDS or if they are HIV negative individuals. And that can be for...
So I guess I'll take a stab at it. I think for the information that was provided today, all of the studies were looking at people who are HIV negative, but there are NIH-funded networks who do work specifically with HIV positive individuals. The HPTN does have a prevention for positive scientific agenda where we look at HIV positive individuals, and there are also 2 other NIH-funded networks, the IMPACT Network and then also the ACTG. Those are primarily treatment networks. So largely the people that they enroll in those studies are HIV positive.
Great,
can I
thank you, Jonathan.
answer, Rebecca?
Yes, sure.
If I understood the question, there's also some studies that are being done within HPTN that are specifically addressing people who are HIV positive. HPTN 052 that looked at serodiscordant couples was specifically for people who are living with HIV, and there's various tests linked to care studies that HPTN is conducting that also enroll people or are working with increasing testing and treatment among people who are living with HIV.
Hi, this is Claire. I just wanted to add that, you know, the people that actually participate are anyone that's interested. You know, people do have to meet certain eligibility requirements for the study, and it's usually best, or well, it's usually necessary that you're in the location where the study is being conducted in one of those sites that we were all referring to, so that you can come to the clinic and get testing. But we always encourage people to, if you're interested, to be open to it. You know, it's the only way that we get information about these products. It's really the only way to advance this kind of research and science, and all of us, I know, would agree, we're completely indebted to the volunteers in our studies and what they really provide. It's really incredible how people give of themselves to do this, because often, you know, they don't know if this is going to be something available to them in the future or not, but they do it because I think they want to help other people, and it's really great to see that.
This is Jim Maynard. I just add one more thing in there, which will sound kind of sappy, but volunteers are the best people in the world. The majority of them, when we do studies afterwards, have signed up to be in a trial for altruism. It's because they really care about their community, they care about their friends, they care about their family, and that's the reason they volunteer. You know, not everyone can volunteer, but everyone can still be involved. They can volunteer, just not as a trial participant. They could serve on the Community Advisory Board. Every site in the nation has what's called a CAB, where people from the community can sit on it. They can advise those doing the research, give community input. That's always a great way they can get involved as well.
Great. Thank you all for your responses. And I think if you're on the call and you're interested in becoming a volunteer, or learning more about how to become a participant, you can visit each site's or each network's website and get more information about participating in either the CAB or becoming a participant in a trial itself. So the next question that we have is for Claire, I believe. And so it says, if both ASPIRE and the RING study have shown that results effectiveness, or if they show effectiveness, will you still have to wait for the HOPE trial before approaching the FDA for approval of the RING?
You know, I think that's a great question. I'm not really sure how that all plays out. You know, sometimes it depends on what level of effectiveness we find in those studies. So I think it's all going to really depend on what we find. And, you know, sometimes if the evidence from both of those studies, let's say, is very, you know, I'm being hopeful here, is that they were highly effective, then that's an argument to go forward while hope is still underway. So it always depends on different factors, but I think that the idea from the researchers and everyone involved in this is to get a product in the hands of women as soon as possible. So that's what we're seeking, but we'll have to see how it all plays out once we know the level of effectiveness and go from there.
Okay. Great, thank you for that. And here's another question for you, Claire, about vaginal and rectal microbicides. And the question is concerning if we saw that women just didn't accept or didn't use vaginal microbicides because of their male partners not liking the feel of it, then what leads researchers to believe that a rectal gel would be more acceptable?
That's a really great question. So I just want to clarify, we don't really know that. That was just a case study that I shared with you. Not all the women said that it was because their male partner didn't like it. Some of them didn't like it. So I just want to make that clear that there are a lot of reasons out there why women didn't use the gel or the tablets in voice. I think, though, the reason why we think that there might be a better acceptability for a rectal microbe is because lubes are often used during anal sex, whereas, whether you use a lube or not during vaginal sex, it's very individual, it's very cultural. They're not necessarily always used, but a lube during anal sex is more likely, it's more common. So our idea is, boy, if this rectal microbicide could possibly feel like a lube, then would it have a chance to be more acceptable for people during anal sex? So that's sort of the idea behind it. And, just the whole idea of the rectum and the vagina being different and what that all means in terms of acceptability for a product. But that is a really great question, and that's sort of the reasoning why we decided to go forward with the rectal microbicide, is because we started these studies even before we knew the results of voice. So there is that part of it too, but one of the reasons was just the idea of acceptability and how different that might be for anal sex.
Great. Thank you for that clarification for all of us, Claire. And so we have another question and I think this will probably be for Jonathan or Georgette, and this is concerning the PrEP effectiveness. And so one of our callers is wondering why are there differing numbers in terms of how effective PrEP is. He mentioned that there are some doctors who are saying it's 99% effective, but then the CDC uses like around 92 or 96% effective. Why are these numbers different?
So the studies that I talked about, they all reported different numbers. They were based on, they give one number, which is the overall effectiveness for everyone that participated in the study. It looks at everybody, and they come up with a number. But the number that a lot of people are using is the number for people who actually use the product consistently. And so typically those numbers are a lot higher. I think I used in IPREX OLA, I said like 50% effective when you looked at all the participants in the study, but when you looked at those who used at least 4 of the pills, took 4 pills weekly, it was 100% effective. So a lot of times people report numbers for those people who actually use the product and omit the numbers for people who don't use the product. And so that's why it varies. The effectiveness is dependent upon adherence. We know it works if you take it as it's prescribed.
And Rebecca, a simple parallel to think of for most people may be the use of the birth control pill. We know what may be quoted to us for effectiveness for a birth control pill, so that's assuming, as Jonathan pointed out, that the pill is actually taken. And that number is going to vary quite a bit if someone chooses to have unprotected sex and not take the pill. Then, of course, that effectiveness will go down quite a bit. And it's part of that curve that Claire talked about in whether or not people are comfortable or able to take a pill on a daily basis or not. But I think that's a good point. There are some people who have raised objections to PrEP talking about some of those lower numbers, but that's simply a function, as Jonathan explained, on whether or not someone is adherent to the medication. And that becomes a very important consideration, because a lack of adherence on almost any type of daily medication is going to greatly change the effectiveness of that medication.
Can I add something too, Rebecca?
Sure.
Georgette and Jonathan are both exactly right. There is this other concept too about doing these kinds of studies that, you know, they're not exact. We're taking a sample of the population and we're pretty much, you know, extrapolating some of these findings. And it's all done with statistical, you know, methods that I don't actually understand. I've always been awful at statistics. But there is this thing called a confidence interval. Whenever a study result is reported, they always say that they have the confidence that the true result lies between this point and that point. And I think all that gets at is that there is uncertainty in science and in research. And it would be actually really surprising if one study and another study had the exact same results because of this uncertainty. So it's just sort of the way that the research is done. But I think it's a great question, because then you start thinking, well, does it work or doesn't it work? And Georgette and Jonathan brought up those really good points about people and how often they're using the product. But the other part is, too, that, you know, it's science, and it's not always 100% the truth. We try to get at the truth as close as possible, and we do multiple studies to confirm results so that we know that we're on the right track. But there's always that little bit of uncertainty involved.
This is Wakefield. I really appreciate the comments of my colleagues, but I want to also just remind folks that from the earlier question, this is why we gather phase 4 research data. Phase 4 research is about how the drugs work in practical application on a day-to-day basis. And that's why you have demonstration projects. That's why you have education for healthcare professionals. I think with PrEP it's important to keep in mind a few things for all of us, and that is that, and maybe people on this call are not aware of it, but in the last month we've seen two major developments with PrEP. In England, they had a study where they were testing the same PrEP regimen, daily PrEP use, to see if they got the same data in men who have sex with men in Great Britain. And they stopped the study early, or at least they said it is unethical for us to continue to delay giving this drug to people because of the number of infections we're seeing in people who don't have the drug. So we don't have the exact data from that study yet, but we do know that they saw a big difference, a big enough difference to stop the study. In France, there was a study looking to see if we gave PrEP to people only when they had sex. We gave it to them. They took a pill before they had sex and within 24 hours after sex, and with another 24 hours after that. That study, too, was stopped because they said, this drug works too well. We cannot ethically delay giving it to people. So they still might get an answer to the question in France, but they've stopped having it as a placebo-controlled trial because it's working. So people who say we don't know enough about it working are people who are afraid for some reason for this to move forward as an effective intervention. We know for people that are HIV positive, if they don't take their antiretroviral therapy, they don't stay with a controlled viral load. We know for every other drug, people who are diabetic that don't take their meds, they don't control their diabetes. So, that's why I keep working for a vaccine, because once they stick it in my arm, I don't get to go home and figure out whether or not I want to take it. So we're not there yet. So we've got to get these other methods of prevention in the hands of people as quickly as we can.
Great. Thank you, Wakefield, for that additional comment. So it is now half past, so I will, but I think these next two questions are really important, so I'll get to these. So hopefully everybody can stay on for an additional 5 or so minutes so we can get answers to these last two questions. So we had a quick question that was about cabotegravir, and Julie wants to know, what class of drug cabotegravir is, and is it already approved?
It's an integrase inhibitor, and it is not approved for use for treatment or prevention yet. It's just been, they're just looking at it. It's been looked at in clinical, like in controlled scientific environments, in petri dishes. They've taken tissue samples and introduced it. They've looked at phase 1 data, and now they're looking at phase 2 studies. But again, we don't know if it's effective for prevention or treatment.
Great, thank you, Jonathan. Okay, so the last question is also from Julie, and it's talking about the prevalence of HIV in the South, and she wants to know how do the clinical trials networks plan to encourage the development of new trial sites in the South. She said all we see from the slides was UAB in Birmingham, Alabama. I know there was also Chapel Hill, North Carolina, but I'm not sure if there's any other ones that we're using. So can anyone answer if there are new developments or any trial sites that are coming out of the South?
So this is Wakefield. I can speak for you for the HIV Vaccine Trials Network, and I could also tell you that going forward, the VTN and the PTN will be doing studies together, and we're trying to figure out the answer to this question. In the VTN, when we've needed additional research sites in the South, we've gone to people who have expertise doing research, and for HVTN 505, for example, we added sites in Houston, in Dallas, and in Orlando. And we're going back as we move forward with our planning for the next group of studies to look again for a place where there's investigator expertise, there's community engagement by that investigator enough to actually educate people and enroll them so they could make a clear decision, and there's clinical capacity. One of the great things that I think will also be helpful is under the Affordable Care Act, you've got a number of commitments from HHS to FQHCs, federally qualified healthcare centers. And so as we look forward, we actually have a visit in a couple of weeks with one of those in New Orleans. We are looking for sites in the South. We're open to people coming back saying, here's a great site, a great investigator, and introducing us so that we can work with them. That's kind of why my job is external relations, is to help us look for those places that have the capacity and have the community engagement to appropriately do the studies.
This is Claire. I'll just add to that. It's a real challenge for certain for MTN, and as Julie mentioned, we go to the same sites because they're familiar with the research and how to do microbicide studies, which are obviously really complicated. But I'll just reiterate what Wakefield said. I mean, I do hear this, and I always, pass it on to leadership. I think that I would be interested in hearing from anyone who has any sites also that they would suggest that might be good for us to think about. I think it is complicated, though, by the challenges that we face in needing people, especially at a phase 3 level of a trial, having experience conducting those trials. But then you get in this catch-22 where you go back to the same sites because they have experience, and then you don't end up, going to their site. So please do, though, contact me or follow up with me if there's any suggestions you may have, and I'm always happy to bring anything to our executive committee at MTN that comes my way. Thank you for bringing that up. It's a great question.
This is Wakefield. I just want to add one other thing. A number of you have received from the HPTN or from the HIV Prevention Justice Group information about a request for information seeking people to offer before December 1st a response on things they think NIH should do with regard to studies for men who have sex with men. If you think it's important, consider. I'm just making a suggestion. I'm not telling anybody what to do. But, consider if you think this is important, adding that to your comments that you were going to submit before December 1st.
Awesome. Thank you, Claire and Wakefield, for the answer to that question. And I definitely encourage those who are still on the call to make those suggestions. And I'll send an email reminder to everyone who was on the call to kind of submit those suggestions if you have any ideas of places in the South that could or should be hosting clinical trial sites. So I'll go briefly through my announcements. First, which is not on the slide, but first let me thank all of the presenters from today. You gave a ton of information that we kind of will all be using, hopefully. I know I learned a ton, and I hope that our callers actually, also learned a lot about the clinical trials process and what each of these networks are doing and how we can be involved and get that information. So thank you again to all of our presenters for today. Just a quick wrap up. When you close out of the webinar system, your evaluation survey will pop up, so I'm just encouraging everyone to go ahead and take that when it closes. And then also, the last question on the evaluation has to do with the 2015 Brown Bag Lunch program, and so we're starting on that program now, and we want to get your ideas on what topics you want to hear from us for next year. We have a few dates and topics in place already, but we're still looking to fill a few slots, and so we definitely want your opinion on what topics we should include in that. So if you do have any other topics that you want to send to me, please send those. My email is up there, rebecca.i@blackaids.org. And if you're interested in applying for the program, also shoot me an email. And my final announcement is about our 2015 African American HIV University, our AHU, what African American HIV University stands for. So our AHU Science and Treatment College will be launching applications in January. It's a year-long fellowship program that's done in collaboration with UCLA, and it's 4 training modules. And the program is really meant to build the capacity of individuals and organizations that serve Black communities who are either highly affected by HIV or at high risk for HIV. And so if you're interested in that program, we're launching applications in January, so please email programs@blackaids.org and we'll give you the application information as soon as it becomes available. And that's it for us. Again, stay on the lookout for the 2015 Brown Bag Lunch program, and I'm always welcome to suggestions and tips for topics and speakers and things like that. So just shoot me an email at rebecca.i@blackaids.org. And I'll be sure to be in contact with you. Again, thank you to our speakers from HPTN, MTN, MVTN. I'm truly grateful for your participation in this webinar, and I hope you all have safe holidays and eat a lot of good food next week. Thanks. Take care.
Thank you. Goodbye, everybody.
Everyone.